Welcome to Lecture 6 on the topic of soil and plant macronutrients. Nitrogen. This is part of the subject Soil and Science and Plant Nutrition ALM114, which is a component of the agricultural degree. This degree is offered at North Melbourne Institution of TAFE, an education institution based in Australia. For more details about our subjects and educational packages, please visit our website at www.nmit.edu.au. My name is Dr. Nikki Cooley. These lectures were compiled by both Dr. Nicola Cooley and Doug Rao. In Lecture 6, we are going to look at nitrogen function in plants, a little on nitrogen deficiency symptoms. I'm going to talk about the nitrogen cycle, nitrogen pools and processes and fluxes, and end this topic with nitrogen fertilizers. In Lecture 2, you may remember, plant nutrition was defined as the study of chemical elements that are essential for plant growth. For an element to be described as essential, 3 criteria must be met. A deficiency of the element makes it impossible for the plant to complete its life cycle. The deficiency is specific for the element in question. And finally, the element is directly involved in the nutrition of the plant, as for example, as a constituent of essential metabolites or required for the action of an enzyme system. All essential elements that plants require are inorganic. There are 22 essential elements, of which 17 are essential plant nutrients, as defined by Argan and Stout in 1939. Please ensure that you review Lecture 1 on plant nutrition before continuing with this topic if you cannot remember these definitions, or you are not able to recall the various nutrient classification methods that can be used. In this lecture, we are going to use the classification based on the quantity of nutrient that is found in the plant, thus macro and micronutrients. Nitrogen is a macronutrient. Nitrogen is involved in many functions in plants. The first one I'm going to draw your attention to is that of its function and its role in nucleic acids. It is part of the building structure that makes nucleic acids. Nucleic acids are polymetric macromolecules. They are biological molecules essential for all forms of life. The nucleotide monomers, these are molecules that are made up and constructed of a mix of carbon, nitrogen, and oxygen molecules. Adenine composition is shown on the slide on the right hand side. What makes plants unique is that they are able to synthesize their own food, utilizing energy from the sun. Chlorophyll is one such structure that aids in this process. It is called photosynthesis. In the light reaction of photosynthesis, chloroplasts are required. They are made up from nitrogen, oxygen, carbon with a magnesium center. The structure contains a porphynyl ring. This is a stable ring-shaped molecule around which electrons are free to migrate. Because the electrons move freely, the ring has the potential to gain or lose electrons easily, and thus the potential to provide energized electrons to other molecules. This is the fundamental process by which chlorophyll captures the energy of the sun. Chlorophyll and hemoglobin are both metallo-organic compounds. Without nitrogen securing the main ring and holding magnesium in place, the function of this structure would not be as effective. The two processes, nucleic acid and chlorophyll, are very important to plants, but so are some of the other functional components that nitrogen plays a role in. It is essential for all structural proteins. It is essential for enzymes. It plays a significant component to yield. It also can influence sugar content. And finally, it has complex interactions with other nutrients, which need to be considered in its optimal management. Soil nitrogen concentration can be influenced by certain characteristics of the soil. Soil total nitrogen is correlated with soil organic carbon. You will often see soil referred to as ratios of C to N. It is important to remember that nitrogen is a nutrient that is mobile. Therefore, it must be investigated across soil layers and within root zones. Different soil types can influence the concentration of nitrogen. For example, peat soils contain 2.5% or less of nitrogen. Cultivated soils, i.e. agricultural soils, tend to contain between 0.8 to 0.4% nitrogen, while subsoils contain approximately 0.02% nitrogen. When you are requiring information about plant crop use of nitrogen, you must remember to analyse your core samples across the root zones, as the mobility of nitrogen can impact on your results and your conclusions from these results. As nitrogen is important for so many processes in plants, nitrogen deficiency symptoms by experts are considered some of the simplest to identify. Chlorosis is perhaps the most common, as there is no longer enough nitrogen to supply chlorophyll with sufficient quantities. Other symptoms that can be seen are poor plant growth, which results in reduced biomass. Lower leaves, the older leaves, show the symptoms first, since the plant will move nitrogen from older leaves to other important younger ones. Whether deficiencies occur on older or younger plants' leaves is very important in helping determining what that deficiency may be. This is not always the same for all plants. Nitrogen deficiency of tea is identified by retarded shoot growth and yellowing of younger leaves. The physical symptoms that are caused by the numerous other stresses, such as deficiencies in other nutrients, toxicity, herbicide injury, disease, insect damage, or environmental conditions, can all impact on your ability to identify nitrogen deficiencies. The most accurate way to identify a nitrogen deficiency is by conducting quantitative tests in your tissue and assessing these alongside the plant visual symptoms. It is also advisable to conduct soil tests as well. The next few slides will illustrate nitrogen deficiencies in different species, so that you can familiarise yourself with how they present quite differently. This is an experiment on a corn crop, where areas were allowed to become deficient. You can see this close-up structure of the monocotyl leaf and how stripping from the top of the leaf downwards in a chlorosis fashion, i.e. the leaf has gone from green to yellow. The image on the screen here demonstrates a nitrogen deficiency in a red grape. Again, you can see reduced biomass and chlorosis. Now we have a better understanding of how important nitrogen is to the plant and how impacting it is when it's limited. Let us look at how we can manage our soils and our nitrogen so that our plant growth is optimized. This is not simple, for in order for you to manage soils nitrogen, you must understand what happens to nitrogen in the soil. Most of the nitrogen, 97 to 90% in the soil, is tied up in the organic matter and unavailable to plants. Only 2 to 3% is in the inorganic form of nitrate, NO3-, and the substance ammonium NH4+. These forms are available to plants. So you have to start your soil management by understanding cycling of nitrogen. It is a complex process and has several components. It consists of 9 major processes: plant uptake, exchange, nitrification, denitrification, volatilization, mineralization, immobilization, nitrogen fixation, and leaching. All of these processes are illustrated on the slide, and how they interact with each other is also illustrated. Each of these processes, as you can see, can affect what is available to the plant, and hence the yield. It is worth remembering when you are learning about these different processes. Remember specifically how it will impact on nitrate and ammonium in the different soil systems, for these are the 2 forms that are available to plants that affect crop yield, and therefore the most important to you when managing your nitrogen. This figure here illustrates the nitrogen cycle as a full community and includes not only the reactions in the soil, but also those of the crop and livestock that you may be running on your soils. Nitrogen can be found in your plant system as either atmospheric nitrogen or in a solid compound. Atmospheric nitrogen is abbreviated by the formula N2. It composes of 78% of the atmosphere and is biologically inert. That is, without assistance, it is unavailable to the plants to use. As stated in the last slide, nitrogen gas is biologically inert, but it can be converted to biologically active compounds. This is called biological nitrogen fixation. The forms that it is converted to can then become available to plants. There are 3 major nitrogen fixation processes: ammonium fertilizer production, lightning, and biological fixation. Ammonium fertilizers require natural gas, steam, oxygen, and a catalyst to fix nitrogen. Therefore, ammonium fertilizers prices are heavily dependent on the price of natural gas. Lightning also fixes nitrogen, although the amount of available nitrogen that reaches the earth from this method of nitrogen fixation is generally less than a couple of kilograms per hectare. Biological nitrogen fixation is able to fix up to 100 kilograms of nitrogen per hectare per year, a significant contribution to your crop cycle. Free-living cyanobacteria and symbiotic bacteria ribosome are involved in this nitrogen bacteria. The process is an anaerobic one, and it involves the enzyme nitrogenase. Nitrogen plus 8 molecules of hydrogen is converted into 2 molecules of NH3 plus H2. The 2 NH3 is then added together with water to produce 2 NH4. 2 OH molecules are left over from the reaction. The statistics for biological nitrogen fixation are truly impressive. Havin et al. In 1999 concluded from a paper they published that 90 million tons per year of nitrogen was fixed in fertilizers, compared to 145 to 200 million tons per year of nitrogen fixed by biological agents. These are very impressive numbers. Biological nitrogen fixing bacteria are described as disotropes. They are bacteria and archaea that fix atmospheric nitrogen gas into a more unstable form, such as ammonia. A nitrotroph is an organism that is able to grow without external sources of fixed nitrogen. It is not a coincidence that these relationships occur between plants and nitrogen-fixing bacteria. There are indeed sophisticated mechanisms involved. It starts with the plant sending out a chemical signals that attract symbionts or nitrogen-fixing bacteria. Root hairs detect the presence of these organs. They then allow an infiltration thread to be established. Over time, a nodule is formed, protecting the nitrogen-fixing bacteria from other elements, but also allowing the transfer of elements. Nitrogen is transferred to the plant in the form of ammonia, and various sugars are transported back to the bacteria. The sugar composition depends on the species of bacteria. The images on these slides show biological nitrogen fixation. The first is an interaction of photosynthetic cells and heterocysts, and the second is a floating fern that uses bacterial relationship, a symbiote bacteria rhizosome, to fix nitrogen. Two non-biological nitrogen fixation methods occur. That of lightning, which we stated earlier, only a very small amount reaches the plants, and that of acid rain. This is where nitrogen and oxygen form together to form nitrogen oxide, NO-. Nitrogen can be obtained from plant residues breaking down and animal waste. This is in the form of amine groups or proteins and the nitrogen stored in the chlorophyll. Once nitrogen is released in this form, it enters into the soil organic matter. Here it can be converted to ammonium and nitrate, the forms that the plant can then uptake. However, it does need this conversion for it to be able to uptake this way. Many of you will be familiar with the concept of decomposition. This is the breakdown of biological matter. Residues are broken down, and so can manure be broken down. Decomposition is conducted by heterotrophic microbes via respiration. The rate of decomposition is controlled by the oxidative state of the soil, the carbon quantity, and the C to N ratio. The optimal C to N ratio is 30:1. If you retain your stubble, decomposition can occur and nitrogen can enter into your system. Nitrogen can leave your system by being consumed either by grazing animals or other herbivores. As microorganisms decompose organic matter, ammonium is released. In a process called mineralization, the amount of nitrogen converted from organic forms to available forms by mineralization ranges from approximately 15.6 to 74.4 kilograms per hectare per year. Populations of microorganisms in a soil range from 100 million to 3 billion in a gram of soil. Urea is another example of an organic nitrogen and is plant available. Denitrification is increased in soils that have been readily decomposable organic matter because denitrifying organisms rely on organic matter for energy. Plants have been found to increase denitrification rates, likely because of the release of readily available organic matter from the roots and the root tissue. The organic matter, at proper moisture, temperature and oxygen content, is continuously being broken down by the microorganisms and released as inorganic nitrogen into the soil. This process is called mineralization. During the process of mineralization, most of the organic matter is first converted into ammonium, NH4, The process that breaks down the ammonium, NH4+, to nitrate, NO-3, by nitrifying bacteria is called nitrification. This process is very important because nitrate is readily available for use by crops and microorganisms. Nitrates are very mobile in the soil. Mineralization can occur in excess after microbial respiration of carbon. This is why the carbon to nitrogen ratio affects net mineralization. If the carbon to nitrogen ratio is 25:1, this results in net immobilization. If it's greater than 25:1, this results in net mineralization. Wood, lignin, cellulose straw, lucerne hay, compost and microbiomass all contribute slightly different C to N ratios, and therefore will either result in net immobilization or net mineralization depending on the C to N ratio of the material. The top graph on the slide provides evidence for the relationship of nitrogen process and CN ratio. When CN ratio is high, that is greater than 25:1, immobilization occurs. As the CN ratio drops, net mineralization then picks up. As these processes change over time, you will also note that ammonium concentration changes over time. It starts up high during net immobilization, then drops up low, and then is returned to a higher state upon net mineralization. The CO2 concentration follows the inverse of the ammonia relationship. That is, when ammonium is high, CO2 is low, and when ammonium is low, CO2 production is high. Now look at the compound ammonia, NH3. This is a gaseous compound, and it can be volatile, especially at high pH. Ammonia volatilization refers to the loss of ammonia as a gas into the atmosphere and can be a source of nitrogen loss. The process is increased at high pH because ammonium will more easily convert to ammonia at high pH. Therefore, fertilizers that increase pH further, for example urea and anhydrous ammonia, may increase ammonia volatilization. This is less of an issue in well-buffered soils because the fertilizer cannot increase the pH as substantially as in poor buffered soils. The buffering refers to the soil's ability to resist change. For example, clay soils are generally better buffered than sandy soils, and calcareous soils are generally highly buffered. Because ammonia needs to be in contact with the air to volatilize, incorporating ammonium-based fertilizers into the soil will also substantially decrease volatilization potential and increase yields. Volatilization increases with increasing wind, increasing temperature, soil coarseness, and nitrogen fertilizer applications. Cool temperatures and generally well-buffered soils may be two reasons that in areas where these occur, losses of surface-applied urea can be observed. Applying anhydrous ammonium in very wet or very dry soils can increase volatilization because the soil will not quickly seal behind the injector knife, allowing the vapor to escape. Volatilization has been observed to occur the slowest between 15 and 20% in a loam soil. Applying ammonium-based fertilizer immediately before a rainstorm can help push it further into the soil profile where it is less available for volatilization. The best methods to decrease volatilization are to incorporate fertilizers applied during calm and cool periods, and if possible, use split applications to decrease application rates. Keep in mind that ammonium converts to nitrate. nitrification in hours to weeks, and once nitrogen becomes nitrate, it can no longer volatile. One of the products we looked at in the Yang Ying teaching and research demonstrator was the addition of urea and green urea. Green urea is a product that is being developed in order to help reduce the compacting issues of volatilization. Cation exchange is an exchange between ions, charged molecules, that are attracted to the soil's surface. Basically, clay generally has a negative charge, while ammonium generally has a positive charge. Therefore, it will be attracted to the weakly held clay particles. The general term for this process is sorption, S-O-R-P-T-I-O-N, which prevents ammonia from moving very rapidly through the soil. Although it may seem that the ammonium would not be available for plant uptake, ammonium can move away from the soil surface as ammonium levels decrease in the soil due to the process known as diffusion. Hence, there is an exchange of ammonium between soil and soil solution. More ammonium is held by high pH soils, and conversely, ammonium moves more rapidly in low pH or acidic soils. The two negatively charged nitrogen forms, nitrate and nitrite, will be repelled from the negative charges of the clay surface and are not attracted very strongly to the fewer positive charges on the clay surfaces. Therefore, nitrate and nitrite both have relatively high mobility, meaning that they can move easily through the soil and do not undergo much exchange. In addition, nitrate requires more energy to the crop after it is taken up, because the nitrate must be converted to ammonium in the plant before it is made into proteins. Unfortunately, in well-aerated agricultural soils, ammonium readily converts to nitrate in a process known as nitrification. Nitrogen is taken up in either an inorganic nitrogen, rarely organic, for higher plants as NH4 and NO3, ammonium and nitrate, and in microbial communities, the microbe draws down if the C to N ratio is high. Soil ammonium, NH4, can be quickly converted to nitrite, NO2, and then into nitrate, NO3. This process can occur quickly, within hours or over weeks, and is known as nitrification. It only occurs in the presence of oxygen, so it generally will be slow or non-existent in waterlogged anaerobic soils. Notice that the nitrogen in the NH4 loses hydrogen ions, thus lowering pH and gaining oxygen during nitrification. The microorganisms that convert ammonium to nitrite and then to nitrate are called the nitrifiers, or neutrifying bacteria. The second part of this process, the conversion of nitrite to nitrate, occurs very rapidly in all but saturated soils. This is fortunate, as nitrite is toxic to both plants and animals. Nitrification occurs most rapidly at pH levels between 6.5 and 8.5, at soil temperatures between 25 and 45 degrees C, and at moist contents above that of wilting point. Nitrification occurs rapidly in most agricultural soils because they are generally well aerated, near neutral pH, and have warm temperatures. Interestingly, anhydrous ammonium fertilizer undergoes nitrification somewhat slower than other ammonium fertilizer materials because the high pH and ammonia in the band inhibited the nitrifying bacteria. It would be desirable if nitrification occurred more slowly, because nitrate could be leached out of the soil profile, whereas ammonium generally stays in the soil profile and is readily available for plant uptake and utilization. Nitrate is anionic and has a low affinity with cation exchange, that is, it's leachable. Denitrification is the process where nitrate, NO3, becomes nitrogen gas, N2. It is the opposite of nitrification in that oxygen is removed rather than added. Denitrification requires the absence of oxygen or anaerobic conditions to perform. Poorly drained soils can result in a 4 to 5% nitrate loss per day, possibly causing substantial yield losses. This was demonstrated by Hoft et al. In 2000. Similarly, nitrification and microbes are responsible for denitrification, and therefore it occurs faster in warm, moist soils. If you recall, nitrification, that nitrate can only form in the presence of oxygen, whereas denitrification requires that nitrate be present and there be no oxygen. Therefore, denitrification losses of nitrogen are most significant when soils alternate between aerobic conditions and anaerobic conditions. In fine textured soils, this could occur in a flood-irrigated field. It can also occur in fields with shallow groundwater tables, especially during the irrigation cycles or oscillating dry and wet periods. Interestingly, denitrification has been found to occur in soils containing 5% oxygen. Air contains about 20% oxygen. How this is possible if denitrification organisms require anaerobic conditions? The answer is that there are small pores within the soil structure that can be saturated and anaerobic. These anaerobic microsites or microcosms have been found to result in substantial levels of denitrification even in soil surfaces by Halvan et al. In 1999. The amount of denitrification in many soils, however, is just simply not known. Denitrification is increased in soils that have readily decomposable organic matter because denitrifying organisms rely on organic matter for energy. Plants have been found to increase denitrification rates, likely because of the release of the ready available organic matter from roots and root tissue. Denitrification increases with temperature between 20 and 40 degrees C and is relatively constant above 40 degrees C. It is inhibited at pH levels below 5.6 but is relatively constant from pH levels 5.6 to 8. Using practices that prevent waterlogging soils is probably the best way to ensure that nitrification losses are kept to a minimum. Let us think of this system as a mass balance in nitrogen, or a summary of the inputs and outputs. So the nitrogen gains include the exchange sites, mineralization, and biological nitrogen fixation, while the losses of nitrogen include plant uptake, absorption to exchange sites, denitrification, volatilization, and immobilization of the nitrogen pool. When your losses equal your gains, then the system is at equilibrium. If, however, you have more losses than gains, you do set up the potential for leaching. Leaching is the loss of the substance from your ecosystem. Nitrate is highly mobile. The areas with the highest risk for nitrate leaching are associated with high precipitation, irrigated conditions, or coarse-textured, shallow soils. Nitrate leaching, like denitrification and volatilization, represent an economic loss because once the nitrogen has left the soil system, it is not available for crop uptake. In addition, there is a health concern that you must be very aware of as farmers. This nitrate can get into the groundwater. Nitrate concentrations are regulated by the EPA in Australia. Different countries have different organisations. If the average nitrate levels in groundwater approach the maximum contaminant level goal, or MCLG, which is 10 mg per litre or 10 parts per million, action will be taken by the EPA, and this can include legal action as well as fines. The factors that contribute with high groundwater nitrate concentrations include coarse soil textures, low slopes, dryland cropping production, tilled cropland, and summer fallow. The lack of nitrogen and water uptake during summer fallow periods likely increase nitrate leaching. Practice that increase crop uptake and decrease excessive percolation should minimize the amount of nitrate leaching. Nitrate can also move upward, especially in semi-arid and arid regions. Upward movement of nitrate and soluble ions occurs when evaporation exceeds precipitation, causing water to move upwards. The ease that nitrate moves, either upwards or downwards, affects soil sampling methods for nitrate. Please ensure that any sampling that you do includes the movement of nitrate. Now let us review nitrogen fertilizers, of which there are many on the commercial product. Urea is perhaps the most common and contains 46% nitrogen. The advantages are that it's inexpensive, it's water soluble, so can be applied as a fertigation as well as a fertilizer, and has a high nitrogen content. Disadvantages of using urea: that it is susceptible to volatilization of NH3, it can burn seedlings, and it can raise soil pH. Ammonium nitrate contains 50% nitrogen. It is more stable than urea. It is a slight higher transport cost than urea. Calcium nitrate contains 15% nitrogen. Calcium for the flocculation of clays on sodic soils. Its disadvantage is that nitrate leaching potential, and it has storage issues. Please be aware, if you purchase any nitrogen fertilizers, that you are familiar with the regulations. If nitrogen is above 45%, then you need certification to use and to carry these fertilizers. Ammonium sulfite provides 21% nitrogen, but also includes 24% sulfur. The advantage of this fertilizer are that it's easy to handle, it can be used in low pH soils, and it retains NH4. The disadvantage is its low nitrogen, high salt, it can burn seedlings, and it is expensive. Monoammonium phosphate contains 11% nitrogen and a range of phosphorus between 48 and 55%. The advantage is that it's a source of phosphorus when both elements are deficient. The disadvantage is that the concentration of nitrogen tends to be low, and nitrogen is somewhat volatile in this fertilizer. Diammonium phosphate contains between 18 and 20% nitrogen and 48 and 54% P. The advantages of using diammonium phosphate are that it's a source of phosphorus as well as a source of nitrogen. The disadvantages of using diammonium phosphate are similar to that of monoammonium phosphate, in that the nitrogen level is low and it's somewhat volatile. This brings us to the end of this lecture on nitrogen. In this lecture, after listening to it, I hope that you have an understanding of the role and the important function of nitrogen in plants. In order to manage nitrogen optimally in your farming system, you need to understand the nitrogen cycles. These can be thought of as nitrogen gains and nitrogen losses. You can get nitrogen gains by fertilization, manure, release from the exchange sites or cation exchange, the process of mineralization, and biological nitrogen fixation. The losses to the system are plant uptake, sorption to exchange sites, denitrification, volatilization and immobilization. And finally, we ended this lecture with a review of some of the commercial nitrogen fertilizers available on the market. Please ensure that you are familiar with your legal EPA requirements for Australia and nutrient handling requirements.